All right, let's dive into something pretty amazing. There's this biological copy machine, and it's working inside you right now, this very second. It's the invisible force that's building you, repairing you, and basically keeping you, well, you. I mean, have you ever really thought about it? You get a tiny paper cut, you forget about it, and then a few days later, poof, it's just gone. How does that happen? Or get this, every single one of us started out as just one cell, a single fertilized egg. So how in the world does that one tiny cell turn into the trillions of cells that make you, you? Well, the answer to all of that is a process called mitosis. And it's this tiny microscopic marvel that lets one cell split into two perfect identical copies. You can really think of it as the engine that drives all of life's growth and repair. So why should you care? Why does this matter so much? Well, for starters, it's literally how we grow up. It's how you go from a baby to an adult. It's how your body patches itself up when you get hurt. And get this, it's happening all the time, replacing billions of old, worn-out cells every single day. We're talking everything from your skin cells to your blood cells. For some organisms, it's even how they reproduce. So yeah, it's kind of a big deal, absolutely essential for life. Okay, but here's the thing. A cell can't just decide to split in two whenever it feels like it. Nope. Before the big show can even start, there's a whole lot of prep work that has to happen. See, mitosis is really just one part of a much bigger story, something called the cell cycle. You can think of the cell cycle as a three-act play. Act 1 is interphase. This is the long, behind-the-scenes preparation stage. Then you get the main event, Act 2, mitosis. This is where all the genetic stuff gets divided up perfectly. And for the grand finale, Act 3, you have cytokinesis, where the cell actually splits right down the middle. And what's really wild is how much time a cell spends just getting ready. Interphase is, by a long shot, the longest part of the whole cycle. It's like the cell's day job, you know? It's just living its life, growing, doing all its normal cell things, while also meticulously getting everything ready for that big split. And this prep work is no joke. The cell has a super important checklist it has to get through. The absolute number one thing, it has to make a perfect copy of its entire DNA blueprint. Plus, it duplicates all its important machinery, the organelles, and it loads up on energy, kind of like carbo-loading before a marathon, to power the whole division process. All right, so once all that prep work and interphase is done, the main event can finally start. And mitosis itself, it's like a perfectly choreographed dance in 4 steps. The whole point is to make sure that copied DNA gets sorted out with absolute precision. So the music starts with step 1, prophase. Picture this. All the long, messy, spaghetti-like DNA inside the nucleus starts to wind up, coiling super, super tight until you can actually see these distinct X-shaped things. Those are the chromosomes. And while that's happening, the wall around the nucleus, the nuclear membrane, just dissolves. That clears the dance floor for the next move. Okay, next on the dance card is metaphase. The easy way to remember this one is M for middle, because that's exactly what happens. The chromosomes do this incredible thing where they all line up, single file, right smack in the middle of the cell. It's like a perfect honga line, and getting this alignment just right is totally critical. And now for the most dramatic part of the whole dance, anaphase. Just think A for apart. Because in one sudden, perfectly timed move, the 2 identical halves of each chromosome, they're called sister chromatids, get yanked apart. Tiny little fibers pull them, and they race towards opposite sides of the cell. This is it, the big separation moment. And to finish the dance, we have telophase. So once the chromosomes get to their corners of the cell, new walls, new nuclear membranes start to build up around each set. The chromosomes can finally relax and unwind back into their stringy form. And just like that, the cell has 2 identical command centers. So the dance is over, the nucleus has divided. But wait, we're still looking at one giant cell that just happens to have 2 nuclei inside. The job's not quite finished. We need that final act, the actual splitting of the cell. And that final step has a name, cytokinesis. This is where the cytoplasm, you know, all the jelly-like goo and the organelles floating around inside, gets divided up. The cell membrane starts to pinch in from the sides, squeezing tighter and tighter until, pop, it separates completely, and now you have 2 separate cells. Now, this is actually where we see a cool little difference between animal and plant cells. In animal cells, like ours, the membrane just pinches in. Easy enough. But plant cells have that tough, rigid cell wall, so they can't just pinch. So what do? They build a brand new wall, called a cell plate, right down the middle, basically building the divider from the inside out.
So, after all that, what's the final score? Well, you started with one parent cell, and now you've got two daughter cells. And these aren't just similar. They are perfect, identical, genetic clones. Each one has a full set of 46 chromosomes, exactly like the cell they came from.
Now, it's really easy to get mitosis mixed up with its cousin, meiosis, so let's clear that up. Mitosis, what we've been talking about, is for growth and repair. It makes two identical body cells. Meiosis, on the other hand, is only for sexual reproduction. It makes four genetically different sex cells, and they only have half the chromosomes. So, yeah, two totally different jobs. This whole process is so elegant, and almost all the time it's completely flawless, but you have to remember, this is a really high-stakes operation happening inside our bodies. So what happens when the perfect copy machine messes up? What happens when it loses control? See, this cell cycle has all these built-in checkpoints. They're like little quality control inspectors, making sure everything is okay, telling the cell when it's good to go, and just as importantly, when to stop. But what happens if those stop signs, those critical signals, get broken and the cell just ignores them? The result is something we all fear: uncontrolled, runaway mitosis. That is the very definition of cancer. The cell's copy machine is basically stuck in the on position, and it just won't shut off. So how does this happen? Well, it starts with mutations, basically damage to the DNA. And that damage breaks the exact genes that are supposed to control the cell cycle. The cell goes deaf to the stop signals and just starts dividing over and over and over, piling up and forming a mass we call a tumor. And because of this direct link to cancer, figuring out the rate of mitosis is a huge part of modern medicine. Doctors, specifically pathologists, use something called the mitotic index to measure just how fast cells are dividing in a tissue sample. The way they calculate it is actually pretty simple. They look at a sample under a microscope, count up the number of cells that are actively in mitosis, and divide that by the total number of cells they see. If that number, the mitotic index, is high, it's a major red flag that a tumor is aggressive and growing fast. So when you think about it, mitosis has this incredible duality, right? It's the very process that builds us, heals us, and keeps us going, but when it goes wrong, it can be the source of a terrible disease. And that leaves us with a really big question to think about. How do we protect this fundamental process? How do we safeguard this biological copy machine that's at the very core of who we are?